Hello, and welcome to FirstThought Focus. For those of you who are joining us for the first time, FirstThought is a life sciences expert network with a mission to bring subject matter expertise back into the expert network industry. FirstThought Focus is our virtual conference series that aims to showcase the breadth and depth of FirstThought's experts and clients, while also covering a wide range of topics that are relevant and important to the life sciences space. You can find replays of our past events and conference recaps on our website, firstthought.io/focus. Today, we are thrilled to be partnering with KX Advisors to host a panel discussion on what's next in next generation sequencing technologies ahead of next week's ASHG annual meeting. Before we get started, just a quick reminder that we will be taking questions from our listeners throughout today's discussion. If you'd like to ask a question, please post it via the Q&A chat button at the bottom of your Zoom window. And if you'd like to get in touch with us after today's event, you can reach us via email at events@firstthought.io. And with that, I'd like to introduce you to today's moderator, Dr. Brian McNally. Brian is a vice president at KX Advisors who specializes in corporate and commercial strategy, product development, market sizing, and forecasting for diagnostics and life sciences companies. Brian, the mic is yours.
Thank you, Morgan. So I'm Brian McNally, and I'll go ahead and start by introducing KX Advisors. KX Advisors provides growth strategy consulting services to help life sciences marketing, sales, and strategy leaders achieve critical business goals. We do this by conducting comprehensive primary research and data-driven analysis of all stages of a product's life cycle. Our collaborative approach helps us to develop pragmatic solutions for leading pharmaceutical, biotechnology, medical device, health IT, diagnostic, and life science tools clients. Through data-driven insights, our clients receive tools to compete and win across the life science industry. On behalf of KX Advisors and FirstThought, I welcome you to today's webinar, and I'm really excited to introduce our amazing panel to discuss what's next in NGS. To begin with, I'll go ahead and introduce the panel that's here with me, starting with Gosia Jaremko. Gosia is currently the founder of Gosia Genomics, as well as the current genomics laboratory director at the New Hope Fertility Center. As a board-certified medical geneticist, she has extensive experience in directing clinical molecular laboratories, most recently serving as Senior Executive Vice President and CLIA Lab Director of Phosphorus Diagnostics, a genetic testing laboratory. Next is Dale Yuzuki. Dale is a Director of Strategic Digital Marketing Communications at Olink Proteomics. Many of our audience know Dale from his popular life science blog, The Next Generation Technologist, as well as his book, COVID-19 From Chaos to Cure. And most recently has started a podcast, Proteomics in Proximity, talking about the intersection of genomics and proteomics. And last but not least, Brian Krueger is a Vice President of Lab R&D at Everly Health. Brian is a leader in setting up high throughput exome and genome sequencing labs. Up until 2015, Brian focused his efforts in academia, but then moved to LabCorp. In 2020, he led the LabCorp team that developed the LabCorp COVID-19 RT-PCR test, as well as the VIR-Seq SARS-CoV-2 NGS test. Welcome, Gosia, Dale, and Brian. Thank you very much.
Thank you.
So to help get us started, 2022 has been an amazing year in next generation sequencing. There have been several, no less than 3 new platforms introduced to the market with a lot of excitement around changes in cost and sequencing throughput. And I'd like to just go ahead and start with each of your reactions to how these new platforms have come to the market with greater than $2 billion invested in at least 3 of these organizations. There's obviously very high expectations. So, Brian, would you like to go ahead and start us off?
Yeah, sure. No, I think it's a really exciting time to be in genomics. as you said in the introduction, I started my career out in academia. And so, a lot of these activities have been focused on the work at academic medical centers where, these programs could afford to spend, $1,000, $2,000, $3,000 on doing a genome sequence to better understand how genomics interacts with these large populations. And now with the cost of sequencing going down, we can expand these genetic analyses out to much wider populations and much more diverse populations. I think that's one of the biggest keys that we've learned over the last 15 years is that you can't just sequence 8 white people, stick them in a tube and understand everything there is to know about genomics. And so with these decreasing sequencing costs, it really, again, opens up the door to be able to expand these analyses out to much more diverse populations, and for us to really start to key into how genomics affects everyone and not just the people who are involved in these early genetic studies.
And Dale, I know that your blog has covered a lot of discussion around sort of the technical details that sort of empower these different platforms. And I think Brian touched on some of the sort of increase in sequencing. I'm wondering how you see sort of like these new players and how they might have been targeting different applications or even perhaps segments within the marketplace.
Yeah, you certainly have, right? Head-to-head competition. And when you look back, right, people love choices, the ability to choose. And for a long time, I mean, by way of background, I joined Illumina in 2003 when they were launching their first 1152-plex genotyping microarrays. I was in the field selling microarray equipment to the NIH when the Solexa acquisition occurred in late 2006, right? So in 2007, I was selling these Solexa 1Gs. Then joined Life Technologies in 2010 to market and sell the solid platform when the Ion Torrent acquisition happened, 2011. And now, right, there was competition at a certain point, but certainly that NHGRI cost of sequencing decline, that sort of famous Moore's law graph, you see a leveling out last 3 or 4 or 5 years, right? Why? Lack of competition. There wasn't any driver for Illumina to lower the cost per gigabase. Now with, right, this, you mentioned the incredible amount of investment, incredible amount of ingenuity. I mean, we had players, right, that would rise and fall. Genapsys was one of them. There were others, right? Hide and seek from NanoString was a targeted method that was going to revolutionize things in the clinic. Those really have had difficulties. Now we have 3 new players, right? Ultima, Singular, and the third one that starts with an E. Help me with the name. I was going to say Emulate, but it's not Emulate. It is Element. Yeah, Element. And each one, right, it's got particular features and dimensions. Certainly Ultima, looking at the high end, the NovaSeq 6000, and now the NovaSeq, is it the NovaSeq X, right? As yes. far as, where it wants to it's going to compete with, it's going to drive the cost down significantly, right? The NovaSeq 6000 running the S4 flow cell is on the order of what, $6 a gigabase. Now they're looking at with the X chemistry, something like $2 a gigabase. But now I think yet another competitor that's going to be out at ASHG in force, I am sure, is the rebranded, reshined MGI, BGI Complete Genomics. And they've got a T7 sequencer, right? This is the renewed monster instrument that they will have freedom to operate to sell in the Americas as well as the rest of the world, right? And that is going to compete with the Ultima on the high end and the sequencing side. And like Brian pointed out, it's going to be a win for everybody. We're going to see that NHGRI graph take another dive down. How far, how fast, it kind of depends. Ultima plans to launch beginning of 2023. The T7's available today, right? And, the NovaSeq X is going to be out in late 2023. So on the high end, population genomics, whole genome sequencing, varied populations, addressing unmet needs. I mean, it's all, we're in another phase of innovation.
Mm-hmm.
So Gosia, I think what's interesting about what Brian and Dale have outlined so far is there's essentially a number of platforms that are coming on the market. And the theme that so far that we've been talking about is really sort of just let's drive down costs to then open up additional essentially, genomic studies. And I think Brian alluded to it, right? Where it's like, let's not just put, 8 white males together and see how, the genome looks. And with your experience having come from, genetics laboratories, I'd love for you to sort of expand on, how do you see that? How does, price impact, for instance, the studies, that would go on and its interaction within the clinical marketplace?
So I will start with a little bit looking back in 2010 when I was working at Quest Diagnostics. At my business unit, we were doing a lot of Sanger sequencing, and NGS. Other companies were already adopting NGS. We were in the same process, so that was totally new how we apply it. At that time, nobody was thinking about really driving the cost down. So I really love the situation, like they'll underscore that this graph is hopefully will start continue going down, because that's what I'm hoping for. And the reason is for the recent few years. So first in phosphorus diagnostics, right now at Gosia Genomics, the cost of sequencing is very critical when it comes to bringing it to wider population. So when you think about how we function so far, we build all the different databases, but unfortunately they are skewed towards the Caucasians white population. We need to be able to equalize and provide access to everyone. And unfortunately, with the current reimbursement landscape for genetic testing, being in the CLIA laboratory and offering this type of tests where there's really difficult to get reimbursed, and at the same time hoping for bringing the genetic testing to wider populations, this cost going down, it's like very critical. And I'm just hoping it's not only going to be translated in terms of the equipment, and it's great that they are new players. And by the way, elements, technology, and innovation, I think this is like a very important to think out of the box, to think like, hey, like loop-seq technology, right? So it's a long read sequencing. However, you can use the short read workflow, which is very important because there may be some sequences out there that people already have, like mini-seq, which I honestly consider like a very good workhorse for like a targeted panels. And they would like to drive this cost further down. So having those options on the market, it's really critical. Otherwise, we won't be able to bring personalized medicine, personalized genomics to everyone. And I believe that every adult and every newborn should have access to be sequenced and have this information. And obviously, there are some aspects to that, interpretations and classifications of the variants that change over time. We learn more, but the bottom line is we all should have access. Doesn't matter, which populations, and basically having competitions coming on the market and driving in terms of equipment and potentially new options, how you can deliver these healthcare to people. It's very critical.
And so, Brian, I would just add that, I think part of the struggle here with these 3 new platforms, and, Dale touched on this a little bit, is that it's a matter of scale, right? And so Illumina offers products along the whole range from the mini-seq to the MiSeq to the NextSeq to, the Nova 6000 and now the X and the X Plus. And people are really familiar with the Illumina workflow. And so the thing that everybody needs to keep in mind is that the Illumina workflow has been primarily like a manual workflow, and it fits really well within an academic medical center where they have all the smart people that know how to do all these cool, amazing things. On the flip side, if we want to, see really broad adoption of these technologies, we probably need to start thinking about systems that fit very well within the hospital environment. And the group that has really excelled there, and Dale knows this as well as anybody else because he used to work there, is Thermo, right? And so Thermo has their Genexus line now, which is basically a point and click instrument where you take a tube, you put it on the extraction little robot, you put it on the ion chef, and then it goes right on the Genexus and you get sample the answer immediately. The only issue with that system is that it's like $50 a gigabase, right? And so we need to start thinking a little bit more about how we transition into this environment where we go from having all the really smart people that know all of the the upfront automation, all of the informatics, and get it into a system where we can drop one of these systems within a hospital and actually get a $100 or $200 genome out of it. Gosia actually makes another really important point: reimbursement, right? And so, like, if the cost of goods sold for a genome is $100, is that what the patient is going to say? No, of course not, right? Like, so you have to add in all of the, customer acquisition costs, paying your people, all those things. And so, when CMS and payers take a look at these things, you'll see the pay rates for like a genome today is $6,000, right? And so unfortunately, it becomes the similar problem that you see in the pharmaceutical industry, where drugs are $2 million because insurers have said that's what the cost, the value that it provides to them. And so now when patients show up at the hospital and they don't have insurance and they want their genome sequenced, they see a bill for $6,000, not $200. And so we also need to think about how these costs actually impact the patients that we're trying to serve, and think of smart ways to have these reduced costs actually be realized in the people that we're trying to treat.
I really like Brian's emphasis on there's another part to the process, like automation. So many labs, they solely focus on manual labor. And I can tell you, just from the clinical lab perspective, it's very difficult depending on the regions where you are, and it's very expensive to procure the workforce that has experience with. So it's highly sophisticated, it's highly manual. And to be honest with you, on average, like training someone to do the NGS library prep, depending on the process, 2 to 3 days, it could take even up to 2 months to really have a person to be fully, able to go. And in some cases can take longer. And I think this is one of the big barriers of, adoption of genomics. It's not only the cost, it's not only reimbursement. However, the labor also adds to that. So having opportunity to have like a whole workflow where the NGS library preparation is not manual anymore, I think that's going to be critical to scale up, but not only to scale up, to even enter for some entities into this type of testing. And obviously, when I think about barriers, is all the analysis pipeline. So I think what is interesting and what's currently happening, some of the manufacturers, they realize we cannot just give people sequencer, or we cannot just give library preparation kit and here you go ahead and do on your own, because the cost of producing the bioinformatics, the cost of connectivity in order to generate patient reports from the start all the way to the end of the process, including all the lab steps, is just so expensive. Like I can tell you that pretty much for average hospital lab, especially considering lack of reimbursement, those are the factors that prevent people to enter that space also. So I think as we do the innovation on the next generation sequencing, equipment side, analysis side, I think we also need to like think about how put all of this together to make it as solution that will be able to be adapted by regular users.
Excellent point, Gosia, right? Because you're looking at the value chain, right?
Mm-hmm.
From the sort of patient payer, the whole healthcare sort of system on that cost side. And one of the interesting observations, which is basically its history, right? That Illumina is focused on the sequencer piece and has left the library prep piece and largely the analysis piece. I mean, you want to have an interesting discussion, Brian, I'd like to hear your thoughts sometime on base space, right? This kind of sort of a marketplace, right, where people can launch little apps for analysis. But nonetheless, right, Illumina doing the hard work on the sequencer side, but leaving the other pieces for the market. And the flip side is QIAGEN, right, that has library prep reagents on the front end and QIAGEN clinical insights on the back end, right? But they don't have a sequencer, even though the gene reader, right, they gave a go. And then we have, and then everything in between, right? Companies that specialize on the library prep or companies that specialize in the clinical report generation. I can think of many different entities and companies. Some have come, some others have gone, some are still around. But it makes it for a much, it's not just $100 genome, as Brian has pointed out. It's a $6,000 genome because we're looking from tip to tail. We're looking at the entire process. And then you say, well, okay, well, where is what Thermo Fisher has done with the Genexus, right? They've come up with their solution. QIAGEN has a partial solution because they don't have a sequencer, right? They just say, well, we'll just do the Illumina. Is there going to be another player to be able to integrate those pieces? What is going to drive those costs down? Really, these are business opportunities in one sense, right? To be able to take a look at and just say, well, let's go ahead and look at the entire problem, not just bits and pieces.
What is interesting that the newest Illumina sequencer, NovaSeq X, it comes together with Dragon, right? So I think slowly, maybe there is like understanding, hey, like we really need to provide a little bit more. So we are not there yet, but I'm hoping that this is just a first step and manufacturer will start realizing that really in order to bring these to the clinic, they need to do more.
So I think it's interesting because you all have touched on a couple of themes, right? One was the sort of platforms themselves, like just driving down costs and introducing competitors to help drive that. The other was around sort of a standardization, right? That's sort of like Illumina was like sort of the manual that you got trained against, and this is what everybody's got to know to sort of like accomplish this. But the incorporation now of new platforms then sort of diversifies, that training set. It certainly opens up then, are there variations within automation that are then going to have to take hold? And so I'm curious, where do you see those sort of opportunities? Like, what are going to be the challenges as it relates to, for instance, incorporating a new sequencing platform into an automated workflow? Is that going to be new opportunities for things, companies like TCAN and Beckman Coulter and Opentrons, or are those things where it's sort of more of a wait and see how each of these platforms plays out? Same question applies to chemistries as well.
Yeah, it's hard to say, right, which of these will be the winner, right? Whether or not, I mean, nobody gets fired for buying IBM. Nobody will get fired for buying Illumina, right? They're the 800-pound gorilla, and, it is a safe conservative bet. I can easily imagine a Hamilton or a Beckman or an Opentrons, frankly, to be agnostic and just respond to the market signals, which is when they have customers with liquid handling problems and scaling problems, right? The feedback from the Broad with regard to the Ultima instrument is that is it UG100? Anyway, whatever they're calling the Ultima instrument, was genuinely positive. And I'm just trying to imagine a 300 millimeter disc, right, with open fluidics. It just blows my mind in terms of what they've done technically. But nonetheless, to offer, right, a very, very high quality sequence, at least from the data that I've seen. One of the things, though, that goes unsaid is, well, what is the error profile of these new instruments? What is the kinds of bases that you have to be aware of that's unique to that chemistry and that instrument? What kind of data formats, right? Is it going to be a FASTQ file, right? In terms of what everybody understands. And then again, in terms of the error model, when the early days of Illumina, right, people were equating Q scores to the validated Sanger scores, and they were absolutely not equivalent, right? Completely different. I mean, they were called similar, and supposedly it was a Phred score, but it was kind of a signal to noise ratio measurement than anything else. And so, that whole aspect of market information, customers getting their, rolling up their sleeves, making a bet on a new platform that not a lot of information is about, and putting through its paces, it takes a lot of time and a lot of work. And I remember in the early days of Illumina seeing those papers of a lot of hard work with regard to its error profile, as far as the bases that were tricky in terms of all the nuances, the sort of devil in the details that really matter in terms of accuracy and performance.
Yeah, and there's one company that spent the last, 10 years working on their error profile, and that's Oxford Nanopore. So like, it's not a simple problem to solve for any of these people. And that's not to say that Oxford hasn't done some, amazing things with their technology. I think it's a little bit mind-blowing that they're able to like get anything moderately useful out of what they're doing, and they're like actually almost maybe kind of close to being as good as PacBio in another iteration, which like is a little bit mind-blowing and cool all at the same time. But also wanted to touch on the reagent kit manufacturers issue, because that's basically what I spent my entire life doing, was automating these things in a very high throughput way. And so all of the instrument manufacturers that you mentioned, Brian, TCAN Hamilton, Biomech, PerkinElmer, Cyclone, these are all open systems that anybody can program these protocols out on. And if you actually talk to some of the manufacturers, Twist and Roche and all these other guys, they actually have scripts that are written on a lot of these instruments. You'll probably have to tweak them to make them fit your use case. So there's a little bit of technical knowledge that needs to go into that. But it's certainly doable if you have the volumes that justify purchasing one of these high throughput liquid handlers. Roche actually did something interesting recently with the release of their Avenio platform, and it looks like it's built on a TCAN Fluent. And so like you can go with Roche, pick up this TCAN Fluent, and you can buy all of your Roche library prep and capture kits. Do you want to use a Roche capture kit? Maybe, I don't know. I haven't tested the most recent iteration, but it's certainly an option. And then somebody in a Roche hat shows up when things break, and you don't have to deal with it yourself and do all that troubleshooting.
And wasn't that a follow on what Agilent did, right? With did they have automation around SureSelect, right?
I don't know if I would call the Bravo automation, but
they called it a Magna. Anyway,
certainly an option.
But again, an idea of a Bravo with the reagent manufacturer on the SureSelect side. So it's similar to what Roche has done with their.
Well, I don't know if Agilent's changed the Bravo, but previously when that thing broke, you had to ship it back to Germany and lose it for like a couple of months. So like certainly not the most reliable situation for running in a high throughput laboratory. You basically need two to make sure that you have something to actually run while it's
so what I appreciate
getting repaired.
What I love, Brian, is that you're speaking with the voice of authority because you've been there. You suffered through that.
I heard that has happened previously. I'm not certain.
So I think this is very interesting, because the main question is like, who can really capitalize on this opportunity, right? Like single vendors of specific liquid handlers, but also, for instance, Twist Bio with the reagents, NGS library preparation kits, they also provided support to implement some of the scripts on and of those instruments, right? So it kind of like, I feel this is untapped opportunity on the market for someone to actually streamline that or start providing some solutions, because basically the choices are like, there's a lot of different automations out there, but I'm looking from the basic lab point of view. When you want to access this technology, and upfront you need to have some specific costs, not only to purchase the main testing platform, but also account for the validation studies, bringing all that talent to be able to run the test analysis. And then on top of that, also the automated platform, that's becoming very expensive to average user, right? And that's where like my mind is always how we can make this available to everyone, how we can make sequencing a standard of practice for clinical labs and for physicians.
No easy answers there, right? Because we've been kind of waiting for, right, this sort of maybe a single cartridge. Remember back in the day, right? There were ideas around microfluidics and doing a single cartridge of library prep and sequencing. You remember Illumina buying, was it Advanced Liquid Logic? Remember that? And it was a failure, utter failure, right? In terms of being able to automate library prep and make it a more streamlined solution. We're still waiting for this sort of opportunity to be filled. With so many fragmented liquid handling, I mean, I'm curious, Brian, from your perspective, why is the liquid handling market so fragmented? Meaning normally in a given market, you'll have, 2 or 3 major players, but we have what, 6?
Yeah, well, I'd say that owes to the complication of the sample volume issue of, a reference lab, right? Does it make sense to automate something if you're only running 6 samples, which was the problem that Illumina was trying to solve with ALL was, okay, the hospital lab only has 6 or 8 samples, right?
Yep.
Get a
Yep.
small, low throughput instrument that can handle that. And so like when you talk about doing an installation of a Hamilton or a TCAN or even a Cyclone, you're talking like 96 sample runs like all day long. So it's like a huge jump in the volume that you need to think about for processing. And once you start talking about processing 96 samples per run, you're at such a high level of scale that you have all the smart people that you need to make that happen. Right?
I see.
Like you're not thinking about this small little desktop instrument to process 8 samples. But I agree with you. I think there is an opportunity there to do this, but does it make sense to do that when it would make more sense for somebody like Element or OmniSeq or one of these others to develop their own little box to process the samples for themselves? Because honestly, if they want to be able to compete with Illumina, that's what they're going to have to do. They're going to have to think about this from the Thermo Fisher model, right? Look at it the way that Thermo looked at it, and know that if you're Element and you're at $5 a gigabase with a tabletop sequencer, you could crush Thermo tomorrow, right?
Yep.
You just have to follow their playbook and execute on it well.
Yep. And that's a lot more moving parts, right? Where the sequencer was hard enough, new chemistry, new instrument, and then to think, okay, well, what about these other missing pieces, right? Mm-hmm. Because the analysis is not a trivial task, reporting not a trivial task, right? Mm-hmm. The reagents and the instrumentation, not a trivial task. But nonetheless, right, like you said, the playbook is there, right? The value chain, right, from sample to answer, as Kaijin likes to say, is complete solution, right? That's what people want. It's like, I want something from A to Z. I don't want to have to put together my own, right, bespoke custom thing with all of its iterations and the in-between.
That's my favorite thing to do, though. Like, I love doing that. It's like going shopping with other people's money. It's so much fun. It's like, I'll go to Watchmaker, and then I'll buy my oligos from IDT. and I'll get my captures from Twist, and I'll get them all arguing with each other to get down to the lowest cost. Like, I love that stuff. That's the best part, right?
Oh,
So,
man.
I mean, you run into problems when you start thinking about sole sourcing it, right? Because then you get into the situation that everybody's in with Illumina, where they control the costs at every level.
Yes.
And like, what are you going to negotiate against Illumina? Good luck with that. Like,
Yeah.
been there. No, thank you.
Yeah. Interesting. Oh, you're going to say
ahead,
something,
Gosia.
Gosia?
So I was going to say that unfortunately, like against challenge for those new players on the market could be sole source, and what that means for clinical lab. You know, how I can trust that if I go with that vendor, how long are they going to be on the market, right? So that's frequently part of evaluation when you decide on like purchasing a platform. And unfortunately, many people just default to Illumina. They've been so many years, they have all the different options. So that's why perhaps, again, like coming back to providing maybe more complete solution could be that factor that could push that forward and say, hey, like we still can go with this new vendor because basically our cost of bringing this in-house is just not going to be as much as, you know, buying Illumina sequencer and figuring everything out afterwards.
You know, Brian and Gosia, for a small amount of time, I was involved in a company that wanted to go cancer early detection. And I was involved in the business development piece of licensing all the bits and pieces that went into the assay. And it was such an eye opener. One was just how some companies had a chokehold, right, on the availability of a critical enzyme, and they could just charge you an arm and a leg and your future children. The other piece of it was, in this world today, how much is outsourced and how you can outsource so much of what you used to hire people for. From it was just wild. I mean, on the contract manufacturing side, on the developing a clinical assay side, on the QC and QA, I was amazed how many businesses, small little businesses I've never heard of, but are offering services to help the development of clinical assays. It was amazing. And I think that that's where an opportunity lies, right, for either new companies that are starting up that don't need to hire a lot of people, as well as from the supplier side of thinking of, well, yeah, how do we help serve this market better? How can we make it easier to develop and validate laboratory developed tests, right? To spin up, you know, either consultants, or specific offerings. You know, you mentioned liquid handling automation, right? You know, could I rent expertise to help automate that process instead of depending on a vendor like Tecan that's worried about like 6 different enrichment platforms, right? Could I hire an expert who could just solve my problem, right, on a per hour basis, and then good day kind of thing?
Mm-hmm.
So there are a lot of those services that exist now. I think, you know, the only thing that I would caution anybody who's starting and thinking about going into genomics and using one of those services is make sure you're going into this for the right reasons, right? Like, remember that at the end of the day, every day, it's a patient that's at the end of that result. And so when you go out and you hire one of these services who just comes in and does everything for you, and you don't know what you're doing after, you know, they walk out the door and cash their check, right? Like, make sure you're being a good steward of laboratory testing. Make sure that you've hired the right people to be able to track the results that you're getting, to understand all the failure modes, to fix all the problems that you're going to run into. It's awesome to get a quick head start using a consulting service. It's not awesome if you're putting out results that are, you know, utter trash and impacting the lives of people who think they're getting a high quality result and aren't.
Doesn't that mean a lot of risk for trying to apply some of these new platforms, knowing that sort of their service and support models are not tested?
Yes. Yeah,
I mean,
but Dale, you know as well as I do, that's not going to stop anybody from purchasing those systems and trying to do something with them, right? And
Sure.
so that is always going to be my argument, that if you're going to get into genomics and you're not going to buy a system from somebody like Thermo, who's again going to send somebody out with a hat to come and make sure that everything's working, like just do
Yep.
us all a favor and stay out of it, because like we don't want to deal with your crappy data.
So I think what's interesting about that is we've started to talk about sort of like the cost of sequencing coming down, and even perhaps opening up the opportunity within, for instance, a given account's wallet to then perhaps purchase in things like automation to kind of make a little bit more seamless workflow. We talked about cost of reagents and some of those additive costs. One of the audience members asked, well, what about things that happened with, for instance, concentration of DNA or library quality checks, and certainly the things that may impact a given sample coming into it, and how we view, for instance, advancements around those type of quality checks, and then maybe how the new platforms or other new technologies have tried to address that.
Mm-hmm.
And so I don't know, have we incorporated that as part of our thinking as far as what's driving cost? Is it wasted time on delivering patient results as examples?
I think this is an excellent question, because frequently when we think about the whole workflow, we concentrate on the main platform and forget that, especially when you talk about the manual library preparation, that there's some ancillary equipment that is definitely needed to perform those pieces. And this depends on the laboratory, how much upfront they want to spend. So some people just go with Qubit, which is very manual. Otherwise, there are some options of multiplate readers like VarioScan from Thermo Fisher, and then there's a range of different bioanalyzers, like different upgrades. Like if you think about tape stations, like a newest version of the tape station, then you also have lab chips. So I personally think the existing solution might be good to for what you want to accomplish when we talk about the manual processes. But again, like my general point is to scale up and to reach the biggest target of like a population screening. Definitely all those pieces would be good if they can come together in a solution that can be fully automated. I don't know, Brian, what do you think about that?
Yeah, no, I think so. What I would say is that there's a range of quality of library prep that you get out of this community, right? And so like the big players who have the smart people that know what they're doing are using like generally the highest quality laboratory preparation methods for creating these libraries. I think there are some quick hacks to getting around some of these issues, like using tagmentation Nextera prep, which, it'll get you a library. Is it the best library that you can get? It absolutely is not the best library that you can get, but it's easy. And it's something that a hospital can perform. Should you be using Qubit or something like qPCR or a droplet PCR based quant before you load your instrument? Absolutely. All of the high quality labs are doing that. Could you use Illumina normalizer beads or normalizer beads from somebody else to do that? Sure, you could, but you're going to pay a little bit more money when those samples are underloaded, and you have to either reload them to fully resequence, or you have to load them on again to top them up to get enough data to do the appropriate analyses. And so I agree with you. I think there's a lot of opportunity here to do something that's high quality, but there are also some options that are available on the market, and one of those is using Illumina's Nextera kits, which I personally don't think have the highest data quality, and you could put together a much more robust system that gets way better data off of your sequencers if you know just a little bit more about how to appropriately do library prep.
Brian, I'd be curious, do you view that then as pretty much a solved problem? That's not the biggest thing to work on in terms of someone who wanted to improve the workflow, that sort of QC of DNA input and the sort of QC of the library prep, these existing methods are good enough?
I think the existing methods are good enough. I just don't think they've been packaged in a way that's easy for, like a small hospital lab to, go out and purchase it and do it themselves without going down, the path that
Yes.
Gosia said, which is the
Yes.
manual strip tubes with,
Yep.
the magnet on the benchtop while you're doing
Yeah.
your ethanol washes. So, I think there's an opportunity here, and
Mhm.
but like I said, Thermo has basically figured that model out for their own system. Now they
Right.
just have to get a sequencer that's like, here in the 21st century.
I no longer work for Thermo, so it's fine when people poke fun at it.
So one of the other things that I think the audience has been asking a lot of questions about is then around this expansion of utility, right? Like we've been talking about driving down costs, and frankly, we're talking about then that opening up automation, which then can help drive down costs and process more samples. If that's the case, then sort of what do we see as then sort of the impact? Are we already seeing like these being widely adopted for more and more population health studies? Or is that sort of still the promise? Is that not been quite realized yet? If there are, please, I'd love to hear some examples, and then I'll have a follow-up question after that around the same theme.
Well, when you're talking about population health, right, we're talking about many, many tens of thousands or hundreds of thousands of samples. And the flagship project is the UK Biobank. And for those not familiar, started in 2006, 2007, 500,000 individuals volunteered to be poked, prodded, examined, full EHR, the consent to get in touch right after, right, an ongoing longitudinal study. And I don't know if you saw the news a couple of weeks ago about 2.5 cups of coffee cutting your risk of all-cause mortality by 27%. Well, that was UK Biobank. They found out who had, brown coffee, decaf coffee, and instant coffee. And fascinating results. Well, the UK Biobank has sequenced all their exomes, and they're about halfway through with their entire genomes of 500,000 individuals. So they have over 200,000 whole genome sequences. So now we're able to do the kinds of studies we can only dream about, right? With not only tons of phenotypic data of all the different vitals, but also all this outcome data, all the drug dosing data, right? Because it's the NHS. Next week's ASHG is packed full of UK Biobank data. And Olink Proteomics has done the Explore 1536 on the 54,000 samples. And there's a lot of data coming out of that. So that's one big population health where genomics is going to pay off in a huge way, right? In terms of variation to disease and then variation to intermediate phenotypes. Not only looking at proteome, circulating proteome as the intermediate phenotype, they're also looking at metabolomics, they're looking at microbiome, all kinds of stuff, right? Other population health is top med, right? This is NHLBI's sort of Framingham study of the future, right? They're looking at transomic for precision medicine. And one of the leaders of top med, Rob Gersten, right? He's looking at proteomics and metabolomics as sort of a real-time readout for cardiovascular health. But top med is one here based in the US from NHLBI. That's population health, it's longitudinal, and it's massive. A third one is million veterans, right? 1 million veterans, longitudinal, captive, right? Single health system, has some unique aspects of it. And a third one, or a fourth one, is all of us. Now, I haven't heard much of all of us, but I know it's still percolating along, right? And this is the precision medicine initiative that Obama announced, 2011. So it's been in the works for a long, long time. But I'd say that, in terms of genomics and population health, and here I gave you 4 examples, 1 from the UK and 3 from the US. That's not to mention there are other projects, the Kadoorie Biobank in China and others. I don't know if others want to chime in here. Gosia, Brian.
Yeah, so I think that one of the first approaches comes to mind is North Shore and Semaphore. I think they launched in 2021 this genomic screening program where pretty much they address cardiovascular, inherited cancers, PGX, and perhaps some other diseases. So I think from my perspective, approach like that, it's very sound, because how many health system we have in US, and basically if North Shore and Semaphore can do it, there's like so much more opportunities to make it more widespread.
Yeah, so I think my only follow-up would be is that, while I think the UK Biobank work is really important, we also need to keep in mind that the UK population is 86% Caucasian. And so like how applicable are the results that we see out of that data set? They're probably really applicable to white people and maybe not so applicable to anybody else that we care about. And so always, whenever anybody talks about the results that they've seen on a population health scale, think about how that study has been ethnically stratified, right? And so think about how those results actually apply to other ethnicities. And that's not to say, again, that what's been done with the UK population is, it's fantastic. It's amazing that the UK government has decided that like it's important to invest in science and do these high throughput sequencing study with multi-level omic data. I'm not sure that all of us quite gets there. They've been parading around quite a bit, talking about how they're going to use Illumina's global diversity array, which is a 3 million SNP array. Like, guys, not quite there yet. Let's start thinking about genomes, because we really need to stop putting all of our hopes and dreams in really bad array data. And then thirdly, I think actually the best population screening stuff is being done in the private sector, right? So like with Helix and Color Genomics and all of these other groups that are now associating themselves with health systems or payers to try to bring this genomic data directly to the employees of employers. And so like when I was at Duke, I gave a presentation every year like on the path to the $0 genome, and it was always because it was going to cost $1 because somebody or the zero, it was going to cost $0 because somebody else was going to pay for it. Like either an employer was going to pay for it or an insurance company was going to pay for it. It's never going to be $0. But
a provocative title, but hey, that's good.
I'm good at those. I'm good at provocative titles. I don't know. Follow me on LinkedIn. You'll see lots of provocation. You get you to click see more.
Brian Provocateur. That's awesome.
Yeah, that's right.
Well, and I think that really leads into the follow-up question, which was that the attendee asked was, okay, there's this population. You started, Brian, to drive it into sort of like individuals. And now when we think about how sometimes we get, like, I'm sure a lot of people have gone through open enrollment, we feel like we're getting locked into our insurance plans and what have you. How do you all see the future laying out with sort of like, okay, I'm getting my genome sequenced. How am I going to get to walk around with it? How am I going to get updates? And how do you all see that moving forward into the future?
So I don't know, Gosia, if you want to go first, because I just got done talking for a little bit. So like, let's hear what you have to say, and then I'll give my opinion, because I have a pretty strong opinion on overselling the genome. But go ahead, Gosia.
Well, you started. I'm very curious now. What is your strong opinion?
No, right. So I think there has been a lot of talk in the last 2 decades about the power of the genome, and I don't disagree that the genome is exceptionally powerful. Is it exceptionally powerful for everybody? It absolutely is not. We all have 5 million variants in our genome. What they do, who knows? Do they do anything in all of us? Maybe not. We don't know. Just because you have a variant absolutely does not mean that you're going to get disease. And so, there was a recent study out of Mount Sinai that did this, that looked for pathogenic variants in an all-comers population. Saw that like 8% of people have a pathogenic variant, and only 10% of those people actually saw disease at any point throughout their life, right? And so you always need to think about how these variants penetrate, right? It's genes and it's an environment. And just because you have a variant doesn't mean that it's something that you have to lose your mind and worry about. And so like, Brian, that's like what we really need to think about. How do you balance having a variant with how you should, approach thinking about it throughout your life? And I really don't think we know the answer to that. And so should you be carrying your genome around and thinking about it all day long? I don't know. I sequenced my exome. There's a bunch of damaging variants in it. And like, what do I do with that information? I don't know. Like, think about like getting colon cancer screening sooner. Maybe.
I agree with Brian's take for the individuals, right? It's really not something that there are compelling reasons for people to get their genome sequenced. All that being said, I mean, when I just got finished talking about the UK Biobank data, the reason that the UK Biobank and 13 pharma companies paid for O-link analysis of 54,000 individuals, which was not cheap, right, was that they're able to use the intermediate phenotype of the circulating protein and the genetic background of those individuals to basically do a GWAS to protein level. And once they do the GWAS, genetic association, to each of the individual 1,500 proteins, they can tie that intermediate phenotype to disease outcome. And what does that mean? It's like a natural clinical trial, where if I'm homozygote for a particular gene like IL-6RA, receptor A, and I have a naturally low circulating amount of IL-6RA, what that means is I have certain risk for certain diseases or certain protection from certain diseases. And thus, the remarkable outcome of this kind of data set and this kind of using a method called Mendelian randomization, a statistical method that associates SNPs or SNVs, like Brian mentioned. Each individual has 5 million of these. It's like, well, Dale, this is a huge data problem. I got 5 million variants per individual. I got 54,000 individuals, and I've got 1,500 protein data points. How do you make sense of all that data? Yeah, computers, right? Just doing pairwise comparisons, grinding through a gazillion comparisons. I mean, my mind explodes when just reciting those numbers, right? 5 million variants, 54,000 individuals, 1,500 protein levels. But as a result, I believe there's going to be a flood of new drug targets. I believe that's going to unleash a torrent of wonderful drug targets. It's already they're rediscovering existing drug targets that they have done via traditional drug discovery methods, and they're going to discover a whole host of new ones. So if I sound excited about the future of genomics, I am, because it's in the population health segment. Now, as far as what the private sector will do with this, yeah, the public aspect is the UK Biobank, and yes, it is for mainly Caucasian individuals, but the disease pathways, I think, are going to hold up. With regard to the private sector, it's going to be really exciting. Go ahead, Gosia.
From my perspective, just, on the clinical actionable side, I believe that we are not ready to interpret the whole genome. So here we talk about situation forward and reverse genetic informations, right? So from my perspective, what is the most actionable is to provide to individuals interpreted data that they can act on. And I agree with Brian that, there are so many genes, disease associations, it's penetrance, it's, you have specific variant. It doesn't necessarily mean that you're going to have that specific disease or all the symptoms. But then if you think about it, it's even more complex than that. It's not only a single gene, a single disease, you also have multiple polygenic risk scores variants. So I believe in general, really to be able to add all of this information together, this is something what we still need to work on and develop. So we don't have those a final prediction algorithms, but still some specific pieces of information that can be interpreted based on the current knowledge. I think it gives many individuals knowledge that access to information and potentially possibility to act on that information. If obviously going through appropriate channels, because the whole situation is much wider than just only getting your genome sequence, then many individuals unfortunately don't get access to genetic counseling, and someone that can help them to understand what that data really means and how they can act on it. So I would say it's a topic for another webinar, because there's so many aspects and angles to that.
Well, Gosia, I appreciate that, because we are coming to the top of our time. I think that, Dale, you summarized it best there at the end, where it's like, this is a very exciting time for genomics and where we're heading. I appreciate each of you as the panelists today. I think that you guys really helped explore some of the topics of like why new sequencing platforms are going to impact the market. But I love the fact that we also looked at the opportunities that were along essentially the workflow, and where that provides both business opportunities for organizations, as well as what it's going to impact when it comes into the healthcare setting. So I think it's been a wonderful discussion. I hope that we get the opportunity to get together soon, and maybe we'll start this one up again. Talk about why maybe I shouldn't have put my sequence on my iPod and walk around back in the day. But there's going to be some questions. Anybody that has questions will try and follow up with you. And we really appreciate all of the audience members' time today. Dale, Gosia, Brian, thank you so much for joining me today for the discussion with KX Advisors and FirstThought.
Great. Thank you, Brian.
Thank you. Appreciate it.
Cheers.